Question 1. A patient presents with symptoms of muscle weakness, fatigue, and difficulty swallowing. A muscle biopsy reveals abnormalities in mitochondrial function. Which of the following organelles is most likely responsible for these symptoms? A. Endoplasmic reticulum. B. Golgi apparatus. C. Nucleus. D. Mitochondria. Correct answer, D. Mitochondria. Mitochondria are the powerhouse of the cell and play a crucial role in energy production. Symptoms such as muscle weakness and fatigue often point to mitochondrial dysfunction, as these organelles are essential for ATP synthesis, which is vital for muscle function and overall cellular energy. Question 2. A researcher is studying the effects of a drug that inhibits the activity of the enzyme DNA polymerase. Which of the following processes would be most directly impacted by this drug? A. Transcription. B. Translation. C. DNA replication. D. Protein degradation. Correct answer, C. DNA replication. DNA polymerase is an enzyme responsible for synthesizing new DNA strands during DNA replication. Inhibiting its activity would disrupt the process of DNA replication, which is crucial for the accurate transmission of genetic information during cell division. Question 3. A patient with a known history of diabetes presents at a clinic with symptoms of excessive thirst, frequent urination, and unexplained weight loss. Lab tests reveal elevated blood glucose levels and the presence of ketones in the urine. Which of the following mechanisms is most likely responsible for these symptoms? A. Decreased insulin production by the pancreas. B. Overproduction of glucagon by the pancreas. C. Increased insulin sensitivity in adipose tissue. D. Enhanced glucose uptake by skeletal muscles. Correct answer. Decreased insulin production by the pancreas. The patient's symptoms, including hyperglycemia, ketonuria, and unexplained weight loss, are consistent with diabetic ketoacidosis, DKA. DKA occurs primarily in type 1 diabetes or severe cases of type 2 diabetes and is characterized by insufficient insulin production by the pancreas. Without adequate insulin, glucose uptake in cells is impaired, leading to hyperglycemia, and the body resorts to breaking down fats for energy, producing ketones in the process, which can lead to the observed symptoms. Question 4. A researcher is conducting an experiment to investigate the effects of a new drug on neurotransmission. They introduce the drug into a synapse and observe that the postsynaptic neuron's response is significantly enhanced compared to baseline. Which of the following mechanisms is most likely responsible for this enhancement? A. Inhibition of neurotransmitter release. B. Increased neurotransmitter reuptake. C. Blockade of postsynaptic receptors. D. Amplification of neurotransmitter signal. Correct answer, D. Amplification of neurotransmitter signal. The observed enhancement in the postsynaptic neuron's response suggests an increase in the effectiveness of the neurotransmitter signal. This can occur through mechanisms that amplify the signal, such as the activation of secondary messengers or increased receptor sensitivity, rather than inhibiting neurotransmitter release, choice A, increasing reuptake, choice B, or blocking postsynaptic receptors, choice C. Question 5. a study investigates the effects of a novel drug on neurotransmission. When the drug is introduced into a synapse, it leads to a marked decrease in the postsynaptic neuron's response compared to baseline. Which of the following mechanisms is most likely responsible for this decrease? A. Inhibition of neurotransmitter reuptake. B. Enhanced neurotransmitter release. C. Blockade of postsynaptic receptors. D. Reduction in neurotransmitter synthesis. Correct answer, C. Blockade of postsynaptic receptors. The observed decrease in the postsynaptic neuron's response suggests that the drug is interfering with the normal neurotransmission process. The most likely mechanism for this decrease is the blockade of postsynaptic receptors, option C, which would prevent the neurotransmitter from binding to its receptors on the postsynaptic neuron and, consequently, reduce the cellular response. Options A, B, and D would typically lead to an increase or normal function of neurotransmission rather than a decrease in response. Question 6. A laboratory experiment involves measuring the rate of an enzymatic reaction at various pH levels. The enzyme's optimal pH is known to be 7.0. As the pH deviates from this value, the reaction rate decreases. Which of the following best explains this phenomenon? A. Changes in pH alter the enzyme's primary structure. B. Altered pH affects the enzyme's cofactor binding. C. pH impacts the enzyme's substrate specificity. D. pH influences the enzyme's tertiary structure. Correct answer, D. pH influences the enzyme's tertiary structure. The observed decrease in the reaction rate as pH deviates from the enzyme's optimal pH suggests that pH affects the enzyme's tertiary structure. Enzymes rely on their specific three-dimensional shapes for catalytic activity. pH can disrupt ionic and hydrogen bonds within the enzyme, leading to denaturation and a loss of enzyme function. This is why option D, pH influences the enzyme's tertiary structure, best explains the phenomenon observed. Question 7. The researcher is studying a population of fruit flies and observes that some individuals exhibit particular traits while others do not. The trait appears to follow a Mendelian inheritance pattern. When two heterozygous flies, AA, for this trait are crossed, what is the probability of their offspring having the same trait? A. 0%. B. 25%. C. 50%. D. 75%. Correct answer, C. 50%. When two heterozygous individuals, AA, are crossed for a trait that follows Mendelian inheritance, the Punnett square predicts the following possibilities for their offspring: 25% AA, homogeneous dominant, 50% AA, heterogeneous, and 25% AA, homogeneous recessive. Since both the homozygous dominant, AA, and heterozygous, AA, individuals exhibit the same trait, the probability of their offspring having the same trait is 25% plus 50% equals 75%. Therefore, the answer is C, 50%. Question 8. A patient presents with symptoms of shortness of breath, chest pain, and fatigue. An electrocardiogram, ECG, reveals abnormal ST segment elevation in multiple leads. The patient's troponin levels are significantly elevated. Which of the following conditions is the most likely diagnosis? A. Pulmonary embolism. B. Myocardial infarction. C. Aortic dissection. D. Pericarditis. Correct answer, B. Myocardial infarction. The combination of symptoms, including chest pain, abnormal ST segment elevation on ECG, and significantly elevated troponin levels, is indicative of a myocardial infarction, heart attack. While other conditions, such as pulmonary embolism, aortic dissection, and pericarditis, can also cause chest pain and ECG abnormalities, the specific combination described is most characteristic of a myocardial infarction. This is a critical consideration in the assessment of patients with cardiac symptoms. Question 9. A laboratory technician is conducting an experiment to investigate the effects of temperature on enzyme activity. They use an enzyme that is known to be active at body temperature, 37 degrees Celsius, and perform the reaction at 3 different temperatures, 10 degrees Celsius, 37 degrees Celsius, and 80 degrees Celsius. Which of the following statements best describes the expected results? A. The reaction rate will be highest at 10 degrees Celsius. B. The reaction rate will be highest at 37 degrees Celsius. C. The reaction rate will be highest at 80 degrees Celsius. D. The reaction rate will be the same at all 3 temperatures. Correct answer, B. The reaction rate will be highest at 37 degrees Celsius. Enzyme activity is generally influenced by temperature, with most enzymes exhibiting optimal activity at a specific temperature. In this case, since the enzyme is known to be active at body temperature, 37 degrees Celsius, the reaction rate is expected to be highest at 37 degrees Celsius, option B. At temperatures significantly lower or higher than the enzyme's optimal temperature, the reaction rate is likely to decrease due to reduced enzyme activity, denaturation, or decreased substrate binding, which is why options A, C, and D are not correct. Question 10. A researcher is investigating the effects of a new drug on neurotransmitter release at synapses. They observe that the drug leads to a significant increase in the amount of neurotransmitter released into the synaptic cleft. Which of the following mechanisms is most likely responsible for this effect? A. Inhibition of calcium ion channels in the presynaptic neuron. B. Blocking of neurotransmitter receptors on the postsynaptic neuron. C. Activation of reuptake transporters on the presynaptic neuron. D. Decreased synthesis of neurotransmitters in the presynaptic neuron. Correct answer, A. Inhibition of calcium ion channels in the presynaptic neuron. Neurotransmitter release at synapses is primarily regulated by the influx of calcium ions, Ca2+, into the presynaptic neuron. When calcium ion channels are blocked or inhibited, option A, it prevents the entry of calcium ions, leading to reduced calcium-dependent neurotransmitter release. This is why option A is the most likely mechanism responsible for the observed increase in neurotransmitter release. The other options, B, C, and D, would typically lead to a decrease in neurotransmitter release.